All right, let's start. So today we are going to finish reproduction and inheritance, hopefully. In the last class, I finished all of this part, Mendelian genetics and chromosomal theory of inheritance. And today we are going to start with molecular genetics, structure and replication of DNA, and hopefully finish replication and gene expression as well. So we did all of this on Wednesday. And so let's start with this. Let's start with nucleic acid. So nucleic acid, remember, the monomers for nucleic acid are nucleotides. Nucleotides are basically two types. Uh, there are, and polymers, remember, polymers are polynucleotides, and polynucleotides are of two types, RNA and DNA. Nucleotides are RNA and DNA when they are single. But when a lot of DNA come together, when you form this double helix structure, that is when you have a polynucleotide. And a nucleotide consists of nitrogenous base, pentose sugar. Pentose sugar is a 5-carbon sugar. It can be deoxyribose or ribose only. If it's ribose, then you'll have RNA. If it's deoxyribose, then you'll have DNA. And you'll also have a phosphate group. Remember, this phosphate group gives the DNA or RNA the negative charge that it needs. And because of this phosphate group, the DNA or RNA is negatively charged. And so yeah, and this is the structure of RNA right here. You can see there is this pentose sugar, and attached to this pentose sugar is a phosphate group right here. And you'll also have a nitrogenous base. And remember that in RNA, you'll have 4 different nitrogenous bases, adenine, cytosine, guanine, and uracil. Whereas in DNA nucleotide, you'll have a ribose sugar, a phosphate group, and a nitrogenous base, but the U would be replaced with a thymine. So you'll have a T instead of a U. And also remember that the difference between an RNA nucleotide and a DNA nucleotide is that a RNA nucleotide has an OH group, whereas a DNA nucleotide has an H attached to the carbon number 2. So moving on, there are two types of nitrogenous bases, the purines and the pyrimidines. And the purines, right? The purines are adenine and guanine. So A and G are the purine, and the pyrimidine are 3 types. You have the cytosine, you have uracil, and you have thymine. So to remember this, I made this acronym. It's called pure as gold. So P, pure, means purine, as A means adenine, and gold, the G refers to guanine. So in pyrimidine, you have cytosine, uracil, and thymine only. And remember that purine bases, adenine and guanines, has 2 rings, whereas pyrimidines, the cytosine, uracil, and thymine, has 1 ring only. And also remember that the purine bases will always bind with the pyrimidine bases. What do I mean by this? The purine base, for example, adenine, will bind with the thymine or the uracil, whereas the guanine would always bind with the cytosine. So A will bind with either T or U, and G or guanine will always bind with the cytosine. And the number of phosphodiester bonds, this I'll explain later, but the number of phosphodiester bonds in DNA or RNA is equal to the number of nucleotides in one strand. So I saw a question in IMAT. There is this DNA structure, right? This double helix, and they give you some bases. So they give you some bases like A, U, T, C. So, and they ask you how many phosphodiester bonds are present in this strand. So in one strand, right? In one strand, you'll calculate the number of bases, and you subtract the total number by 1, and that will give you the phosphodiester bond in one of these strands. All right, so this is how you calculate the phosphodiester bonds in one strand. So uh, moving on to hydrogen bonding. Hydrogen bonding happens between complementary bases of two DNA strands. This is important for the 3D structure of molecules, and many hydrogen bonds will give stability. But if you have individual hydrogen bonds that are weak, for example, when you have A and T, this will have hydrogen bonding, but this individual hydrogen bonding is weak. But when you have multiple nucleotides, when you have a DNA strand, then the many hydrogen bonds that form between this strand would make the DNA strong. It was calculate all bases and -1 for the amount of phosphodiester bond. Yes, if I think I confused you guys here, but remember that the number of phosphodiester bonds in the strand, in the DNA strand, right? If you have a DNA strand, the number of phosphodiester bonds will be 1 minus the number of nucleotides or the bases in one strand. All right. Yeah. So moving on, this hydrogen bonding is very important for DNA replication and transcription so that the strands can be separated. And hydrogen bonds only form between specific bases. So A will only form hydrogen bonding with T or U. And similarly, G will form hydrogen bonding between C only. And uh, hydrogen bond also can easily reform without a chemical reaction. It can be broken by pH changes or temperature changes, and it can also reform without a chemical reaction. So you have two strands of polynucleotides that are anti-parallel, right? And they run in opposite directions. And one strand starts with 5 and ends with 3. And the second strand will start with 3 and end with 5. So when you have DNA transcription, right, this part is very confusing to a lot of people. So when you have a DNA strand that is being synthesized, right, there is the DNA polymerase enzyme. I'll explain the whole process, but just know that the DNA polymerase will always start from the 5 knot and then move on like this and synthesize the new DNA strand 5 to 3. So this part I'll explain again, but the number of purine and number of pyrimidines are the same. So the ratio is always the same in a DNA strand. And yeah, so let's move on to transcription now. Transcription occurs in the nucleus during the S phase of interphase. This is also known as DNA replication. And you need ATP for this DNA replication. And the enzymes, there are 3 enzymes that are needed for DNA replication. The 3 enzymes are helicase. Helicase enzyme breaks the hydrogen bonds to separate the 2 DNA strands. And the second type of enzyme is DNA polymerase, which will synthesize a new strand of DNA. So when the DNA strand wants to form hydrogen bonds, DNA polymerase will form the hydrogen bonds. And another thing DNA polymerase does is it catalyzes the formation of phosphodiester bonds between nucleotides. And it also looks for any errors and repair the DNA code if there is any type of errors in the DNA. And finally, the third type of enzyme is called DNA ligase. It joins the DNA fragments together, and it also catalyzes the formation of phosphodiester bonds like DNA polymerase. So how is DNA replicated? This, remember, is transcription. So the first step is the DNA double helix would unwind. So if I had a double helix, this would start to unwind because the helicase enzyme will start to break the hydrogen bonds and it would separate the DNA. So hydrogen bonds are broken because of helicase and the DNA will unwind, and both strands are used as templates. So remember, when this strand breaks, it would break into two different strands. And each of these strands would be used to make copies of DNA. So you'll use both of these DNA as template strands. Remember this, that one DNA molecule would give you two DNA strands. And both these strands are used as templates. So remember that after the helicase breaks, right? Helicase breaks the hydrogen bonds. Then comes the making of new DNA strands. This happens because you have free activated DNA nucleotides with two additional phosphates. And remember that DNAs have three phosphate groups in total. So the base of activated nucleotides form hydrogen bonds with base on each exposed parental DNA strand. So remember, the free nucleotides in the cytoplasm and nucleus have three phosphate groups, but DNA strand only has one phosphate group. So why does it have two extra phosphate groups? Because these nucleotides are activated with two additional phosphate groups. So when you have two additional phosphate groups, you have activated nucleotides. Now, these activated nucleotides, right? These nucleotides, let's say this is the cytoplasm. These activated nucleotides, the A, U, T, not U, A, T, G, and C, these are all activated by two additional phosphate groups. So the DNA strand, right? This is a DNA strand. It would combine with the DNA template, the DNA strand that was replicated, and you'll form these activated nucleotides will form hydrogen bonds with the DNA strand. And when this happens, the two phosphate groups that were attached, right, they would leave the activated nucleotide. So then you'll only have one phosphate group. So remember, activated nucleotides have three phosphate groups in total, whereas when you form hydrogen bonds with the DNA template, you'll only have one phosphate group on one nucleotide. So yeah. So what happens after you form hydrogen bonds? The DNA polymerase attaches to each of the two separated parental strands and will catalyze the formation of a new strand of DNA. So when these activated nucleotides join the DNA strand, the DNA polymerase would start to catalyze the formation of hydrogen bonds and phosphodiester bonds. So DNA polymerase links activated nucleotides together by removing the two phosphate groups and catalyzes the phosphodiester bond formation. So what is the job of DNA polymerase? It catalyzes the formation of phosphodiester bonds, and it also removes the two phosphate groups. So yeah. And DNA polymerase, again, it also checks for any errors in the DNA and fixes them. And the process continues along the whole DNA molecule. So until it reaches the end, you'll complete the DNA polymerase. The DNA polymerase will reach the end and you'll have hydrogen bonds. So there is a problem with DNA replication because one DNA strand, right? One DNA strand, when you have this DNA template, one DNA strand is synthesized continuously. And this continuously synthesizing DNA is called the leading strand. But the other is synthesized in sections known as Okazaki fragments. What is this? Remember that DNA replication, right? DNA replication, this is the template strand. And the continuous DNA strand that will be synthesized. So the DNA polymerase first moves from the 3 to the 5th. This strand, the 3 to 5, is known as the leading strand. And in the leading strand, you'll have continuous DNA hydrogen bonding because the DNA polymerase will continuously hydrolyze new hydrogen bonds. But this is not the case for the lagging strand or the Okazaki fragments. The lagging strand, which goes from 5 to 3, is discontinuous. So you'll not have in the discontinuous fragment, right? In the discontinuous fragment, which is from 5 to 3, in this type of fragment, it's called the lagging fragment, lagging strand, or the Okazaki fragments. In this type of strand, you'll not have continuous hydrogen bond formation. The DNA polymerase in the lagging strand is unable to continuously form hydrogen bonds. So to fix this problem, right, to fix this discontinuous fragments, you'll have DNA ligase. So what is the job of DNA ligase? DNA ligase, remember, will form hydrogen bonds, H bonds, basically, in the lagging strand, and it joins any nicks together. So DNA ligase will, the DNA ligase is continuously it's continuously forming the hydrogen bonds in the lagging strand. So remember, DNA ligase is very important for the lagging strand because it will form hydrogen bonds in that strand. So at the end of DNA replication, you'll have two DNA molecules, each containing one original strand and one newly synthesized strand. So when I told you that, where is it? I told you that this DNA strand would, this DNA molecule, the original DNA molecule, would separate into two DNA strands. So both of these strands will be called the template strands. And then the DNA replication happens, and you'll have these two DNA molecules. Now, in this two DNA molecules, one DNA strand is called the template strand, and the other one is called the synthesized strand. Similarly, it's the same case for here. I have a question. I thought the ligase synthesizes phosphodiester bonds. Yeah, it does, because remember, DNA ligase has all of these roles as well. It joins DNA fragments together through hydrogen bonding, and it also catalyzes the formation of phosphodiester bonds in the lagging strand. So these two roles are for DNA ligase. Where was I? Right here. So this, at the end of DNA replication, again, you'll have two DNA molecules. So after RNA nucleotides bind to the base of the DNA, the newly formed strand is known as mRNA. This is when DNA replication is done. After DNA replication happens, you'll have something called translation. DNA replication is, remember, it's also called transcription. And translation is when you convert RNA into protein. So protein synthesis is also known as translation. So how does this start? This is basically the summary of DNA translation. It's when RNA nucleotides bind to the bases of one DNA, the newly formed strand is known as mRNA. So remember that this DNA strand that you just made, right, will convert into RNA through DNA polymerase. And this mRNA that you just made will go to the cytoplasm, will move out of the nucleus into the cytoplasm, and mRNA molecules attach to free ribosomes in the cytoplasm. So remember that in DNA, when DNA is made, DNA will convert into something called mRNA. This M means messenger. This DNA will convert into messenger RNA. This messenger RNA which is found in the nucleus, will move out of the nucleus into the cytoplasm through nuclear pores, and then comes translation. So what is translation? Remember, let's start with the translation. Remember that mRNA nucleotides are read as series of triplets. So this right here is one nucleotide. This is another nucleotide. This is another nucleotide. So mRNA nucleotides are read as something called triplets. And each triplet is called a codon. We'll talk more about codon, but let's move on. The mRNA nucleotide sequence is used to determine the amino acid sequence of the polypeptide or the protein. So how do you determine the amino acid sequence of the protein? It's through the mRNA nucleotide sequence. Each triplet in an mRNA is known as a codon. And the start codon marks the site of translation into protein. So how do you start translation? The translation will always start with the start codon. The start codon is the first it's the first set of 3 nucleotides that will come together to start translation. And the stop codon is the site at which translation ends. So the translation ends when the stop codon is introduced. And all of this happens due to something called tRNA. Remember, in translation, you have 2 types of RNA that are used, the tRNA and the mRNA. The tRNA is found in the nucleus, in the cytoplasm. And the mRNA is made in the nucleus and is found in both the nucleus as well as the cytoplasm. Because the mRNA will move from the nucleus to the cytoplasm. So, yeah, and anticodons are triplet bases that are complementary to the mRNA codons. And the anticodons are found on the tRNA. I'll explain all of this, but for now, just know what anticodons and codons are. So anticodons are found on tRNA, and anticodons are complementary to the tRNA, to the mRNA nucleotides. So one tRNA has one anticodon. So remember, one tRNA molecule has one anticodon. And tRNA and amino acids are found in the cytoplasm, as I said. Amino acids are also found in the cytoplasm. So the mRNA nucleotides are read as a series of triplets, as I said. If you have this chain right here, this is an mRNA chain. And remember, mRNA will always be single strand. And so, yeah. And mRNA nucleotide sequence is used to determine the amino acid sequence of a polypeptide. Yeah, I think this is a repeat of what we did. Yeah, this is a repeat. So let's move on to the real stuff. Let's move on to how translation happens. So a few things to note. First is that translation happens in the cytoplasm. And second thing, you have two RNAs involved, the mRNA as well as the tRNA. So this mRNA, right, will have a start codon, and remember that start codon will always be AUG. This is the start codon. AUG or OG is always the start codon, and the start codon will always start with the translation. So mRNA in the nucleus moves out from the nucleus and binds to ribosomes found in the cytoplasm. So mRNA molecule will move on, and this is an mRNA molecule. So this mRNA molecule will bind to a ribosome, and remember that these ribosomes are the ATS ribosomes. The ATS ribosomes are the ribosomes that are found in the cytoplasm, and this is where this is the ribosomes that will cause translation. So mRNA binds to the ribosomes. This is the first step. The second step is that tRNA from the nucleus. Why is it nucleus? It's cytoplasm. Ignore this. tRNA from the cytoplasm attaches to the ribosome. So a tRNA molecule from the cytoplasm will come and bind to this ribosome right here. And tRNA has complementary anticodon. So for example, if the mRNA strand in the ribosome, right, this is the ribosome, if this mRNA strand had AUG, the tRNA strand, right, this is a tRNA, this tRNA will have UAC. So this would be the anticodon. So this is the anticodon that would be complementary to the nucleotides of the mRNA. So remember, tRNA molecules have complementary base pairing with the mRNA nucleotides. So tRNA molecules have complementary anticodons. This is held in place by hydrogen bonds between base pairs of the mRNA and tRNA. So how does tRNA stay fixed in the ribosome? It stays fixed through hydrogen bonds. These hydrogen bonds are formed between the bases of the mRNA and tRNA. So after forming hydrogen bonds, a second tRNA molecule moves into place. So the second tRNA molecule will come into place, and it would form a base pairing with the second codon of the mRNA, or the three nucleotides, right? This will form a base pairing with the second tRNA molecule. So a second tRNA molecule moves into place, and the anticodon on this tRNA is complementary to the second codon of the mRNA molecule. So if I had OG, if this is the first codon, the second codon, for example, let it be UCG, this would be the second codon, and the tRNA molecule will have complementary bases to the mRNA, to the second codon. If this is UCG, the tRNA molecule will have AGC. All right. So yeah, so second tRNA comes into place and forms base pairing with the second codon of the mRNA, and amino acids start to form on the binding site of tRNA and make the protein required. And remember, amino acids are the free amino acids are found in the cytoplasm. So the free amino acids in the cytoplasm, they come attached to the tRNA. The place where the amino acid attaches is called the binding site, and the tRNA will start to synthesize the amino acid sequence. So the amino acid keeps growing, and the tRNA keeps attaching until the protein required is synthesized. It is then released into the cytoplasm to perform its action. So these were the two tRNA molecules. Another tRNA molecule comes, and then the amino acid chain grows, and then this keeps on happening. So new tRNA molecules keep on attaching, and new amino acids are continuously binding to the binding site. And so you're making this big amino acid chain, and this will continue to happen until the protein required is made. And then this amino acid will go through the four structures. So the primary, the secondary, the tertiary, and the quaternary structure. So yeah, this is how a protein is made, and this is called translation. So yeah, this is translation. And we have time, so I don't think we can finish this. So yeah, we'll do this in the next class. Regulation of gene expression. This is where you control how gene is regulated. Gene regulation. What we just did was gene expression. So all of this, remember that translation as well as transcription is known as gene expression. And gene regulation, what is gene regulation? Regulation. Gene regulation is when the genes are regulated and not synthesized. Synthesized, or this is one of the ways gene regulation happens. It's when the genes are not synthesized, or genes are synthesized under protection. So what is the types of protection that that are present in gene regulators? So you have promoters, you have operators. All of these are protections for gene regulation. Gene regulation basically happens so that you get the correct gene to be prescribed. And yeah, so gene regulation, we'll do that in the next class. We'll do all of this in the next class. So yeah, this was today's class. We don't have any questions for today. So yeah, see you tomorrow. Bye. Everything is clear till now, right? Bye.